Okay, I want you to try all these problems by yourself first. This is number 1. Pause the video. Number 2. Number 3, 4, 5, 6. Number 7, 8. And number 9. Okay, before we talk about all these problems here, let's talk about two operons that happens in bacteria. First of all, let's talk about Trp Operon. Trp is short for tryptophan. Since tryptophan is an amino acid that bacteria needs, it's going to keep on making it until it gets too high in concentration. So let's talk about some of the operons here. In operon, which is like a switch that turns on or turns off depending on the different environment, you got promoter here. Promoter section has an operator. Okay, promoter is where RNA polymerase will sit on and it's going to do its job. It's going to start its transcription there. Okay, but it's got an operator section. These are so promoter, operator, and the gene of interest. These are all parts of DNA. Okay, together you call it operon. But this operator section can be bound by some protein called a repressor here. In this case, it's a tryptophan repressor, which is transcribed and translated from this regulatory gene. That's also part of the DNA. So when this repressor is activated, it's going to sit on the operator, which will block the pathway of RNA polymerase. So there will be no transcription happening here. So let's see what's going on when there is no tryptophan. Remember, tryptophan is what they need to make. So when there is no tryptophan, what happens is that RNA polymerase will keep on transcribe these genes that are needed to make mRNA to make the tryptophan. Okay. However, if there's a lot of tryptophan here, look at the bottom one here. The tryptophan itself will inhibit the transcription of its own mRNA. What happens is it's a co-repressor. It binds to the real repressor here, which will activate this Trp repressor. Now, this repressor will go and sit at the operator section. Then what happens? The whole transcription of mRNA to make tryptophan is blocked. So this is nice. When there's a lot of tryptophan, it will block the synthesis of itself. So because tryptophan is present, it represses the production of itself. So this is called repressible operon. Let's talk about the other operon here. This time we have Lac Operon. It's lactose. First of all, lactose is a disaccharide. It's made out of what? Glucose plus galactose. Okay. So when there's a lactose present, the bacteria will make lactase or any kind of enzyme that will hydrolyze this lactose into glucose and galactose. So without any kind of lactose in it, it's pointless to make lactase or any kind of enzyme that will break it down because there is no lactose to start with, correct? All right, so if there is a lactose or allolactose in this case, it's the same thing. It's an inducer now because it will induce or promote the synthesis of mRNA, which will get translated into making more lactase. So when allolactose is present now, it will bind to the repressor protein. Remember, repressor protein is made where? This purple gene, it's called regulatory gene. Okay. And it gets transcribed into mRNA, and that's how repressor protein is made. So when there's allolactose there, it will bind to the protein, not at the active site, right? So allosterically it will bind to it, which will cause the repressor protein to have conformation change. It will change its shape so that it can no longer bind to that operator section. So RNA polymerase is free to make mRNA, to make any kind of enzyme that will break down lactose. So it's pretty neat. Only in the presence of this allolactose will RNA polymerase will then synthesize mRNA to make the enzymes to break down lactose, such as lactase. However, if there's no lactose, look at on the bottom here. If there is no lactose, repressor is active without the inducer here, and it sits there all the time until when? Until allolactose or any kind of lactose is present. So because the presence of allolactose, it will induce transcription. This is called inducible operon. Now let's go back and try the problems. So number one, which of the following statements correctly describes the role of a molecule that controls a repressible operon? What was repressible operon? In the presence of something, it blocks it. What is that? That's Trp Operon. In the presence of a lot of tryptophan, it will turn itself off. Here we go. So the molecule, they're talking about the tryptophan molecule here, binds to the repressor protein and it will activate it. Meaning when the repressor protein is activated, it's going to go into the operon section of the DNA and blocks the whole polymerase. Right here, when the tryptophan binds to the repressor, it will activate it, so it will sit at the operator section, which will block the whole transcription. So number one will be A. Let's move on to number two. The tryptophan operon that we just talked about in some bacteria is a repressible operon. We know this already. Which of the following statements correctly describes this operon? It's permanently active. No, it's not. It will turn itself off when there's a lot of tryptophan. It is activated only when glucose, what? No. It is activated only when tryptophan is present in the growth medium. If the whole operon is activated, it will make more tryptophan even though there is tryptophan is available. So C is not it. C is totally opposite. The answer should be D. It gets deactivated when tryptophan is added, since there's too much. Let's do all these right now. 3, 4, 5, 6. Number 3 says, which of the following is a protein product of a regulatory gene? Where was regulatory gene? Regulatory gene was upstream somewhere. Here we go. Let me show you the picture here. So like this is a regulatory gene, which will make what? Repressor protein, correct? So number 3, regulatory gene makes repressor protein. That was simple. It's something that you just have to memorize. Number 4 would be a lack of which of the following would result in cells' inability to turn off genes. If you don't have this, there's no way to turn the genes off. There will be some kind of repressor because repressor turns it off. And the only repressor here is co-repressor here. So what is the example of co-repressor? Tryptophan. Tryptophan will bind to the actual repressor, which will activate it and represses the whole synthesis of mRNA. Number 5. Which of the following, when taken up by the cell, binds the repressor? What binds the repressor? Well, two things. You got co-repressor binding to it or inducer binding to it. So that repressor no longer binds to the operator. That will be the inducer, correct? When an inducer binds the repressor, the repressor protein changes its shape, so it will no longer bind to the operator, which will turn on the gene. Number 6. Most repressor proteins exhibit allosteric properties, meaning it will bind to somewhere other than active site. Which of the following molecules binds with the repressor to alter its structure? Both the inducer and co-repressor can bind to it to change the shape, but there's no co-repressor here. The best answer will be C. Let's move on. Number 7 and 8. Number 7 first. What will be the result of a mutation that deactivates regulatory gene of a repressible operon? Okay, let's bring that picture back again. Okay, this is a picture of Trp Operon again, which is repressible operon. What will happen if there is a mutation at the regulatory gene? So if this is mutated, then you're not going to have a functional repressor protein. If something's wrong with the repressor protein, that means the co-repressor, in this case tryptophan, cannot bind to it to activate it. So there will be no activated repressor that can block this process. What does that mean? It means there's nothing to stop the transcription of it. Correct? So let me see. You're going to have, it's going to be C, continuous transcription of this enzyme coding gene. Let's move on to number 8. So number 8, which of the following must occur for repressible operon to be transcribed? So this is another repressible operon. So in order to have transcription, you must have RNA polymerase. Yes. And that RNA polymerase must bind to the promoter, and repressor must be inactive. If repressor is activated, you'll block the process. So A will be A. Let's move on to number 9. Number 9, allolactose, an isomer of lactose, is formed in small amounts from lactose. An E. coli cell is presented for the first time with sugar lactose. So you're going to have a lactose. What does that mean? Then the E. coli will make lactase or any kind of enzyme that will break down lactose. Okay, which of the following processes occurs when the lactose enters a cell? So when the lactose is present, right here, allolactose is present, it will bind to the repressor protein, which will make it inactive so that it cannot bind to the operator to block it. Okay, so when allolactose is present, you'll bind to the repressor. Let's see. A, the repressor protein attaches to the regulator. Allolactose binds to the operator. Nope, allolactose binds to the what? The repressor. C, the repressor protein allolactose bind to RNA polymerase. No, they don't bind to the RNA polymerase. D, allolactose binds to the repressor protein. Yes, that must be it. By now, I hope you understand both Lac Operon and Trp Operon. It's not that hard. Just review all the terms and mechanisms there.